Hello everyone, myself Priyanka Kande-Patil, Assistant Professor of Department of Microbiology. So today's topic, that is antimicrobial drugs or antibiotics. Anti means against and biotics means life, against the life. Antibiotics are the medicines which are effective against various types of infections. It depends on chemical structure, then types of organisms, active against bacteria, fungi, yeast, parasites, and actinomycetes. Type of action. Also, it depends upon the mechanism of action. Mechanism of action, that is cell wall inhibitor, protein synthesis inhibitor, cell membrane inhibitor, and nucleic acid synthesis inhibitor. Nucleic acid synthesis, we know that there are two nucleic acids, DNA and RNA. DNA undergo indirect mechanism of action and direct mechanism of action. Also, it depends upon the antibiotics are obtained from which microorganism. For example, bacitracin is obtained from bacteria. Penicillin is obtained from fungi, and tetracycline and chloramphenicol are obtained from actinomycetes. Next, it depends upon the spectrum. So, there are two types of spectrum. One is narrow spectrum and second one is broad spectrum. Narrow spectrum, it is active against either gram-positive bacteria or gram-negative bacteria. And the broad spectrum antibiotic, it is active against both gram-positive and gram-negative bacteria. So it depends on chemical structure, types of organisms, type of action, mechanism of action. Antibiotic are obtained from which microorganism, spectrum, and the last one, mode of action. So mode of action, again it is a bacteriostatic and bactericidal, that is inhibitory mode of action and killing mode of action. First, we see the cell wall synthesis inhibitors. The beta-lactam, it is the first ever discovered antibiotic, and the penicillin, it is the first generation beta-lactam antibiotic. So this group of antibiotic include penicillin, carbapenem, cephalosporin, and monobactam. The mechanism of action. So we know that the cell wall of bacteria is made up of peptidoglycan, and peptidoglycan consists of two subunits, that is NAM and NAG. So NAM, it is N-acetylmuramic acid, and NAG, that is N-acetylglucosamine. NAM, this particular subunit is a pentamer because it is made up of 5 amino acids. So these 5 amino acids include first L-alanine, second one D-glutamic acid, third one L-lysine, and 2 amino acids, that is D-alanine. So these are 5 amino acids. That's why N-acetylmuramic acid is a pentamer. The initial step is the binding of drug to the specific drug receptors. And the specific drug receptors are known as penicillin binding protein. So these penicillin binding proteins are present on the surface of that particular bacteria. This enzyme involved in transpeptidation reaction. So transpeptidation reaction is nothing but the cross-linking reaction. Then we see the resistance. First resistance, that is decreasing influx and increasing efflux. So influx is the entry of drug and efflux is exit of drug. So the first resistance mechanism, that we decrease the entry of that particular drug and we increase the exit of that particular drug. Second resistance, that is by using penicillinase enzyme. Penicillinase, which is also known as beta-lactamase enzyme. Third resistance, alteration of penicillin binding protein. that is a transpeptidase enzyme. So this is the classification of beta-lactam antibiotic. So first antibiotic, that is penicillin. Penicillin, it is the first generation beta-lactam antibiotic. It is discovered by Alexander Fleming in 1928. It is isolated from Penicillium chrysogenum and Penicillium notatum. Also, some Aspergillus species are responsible to produce penicillin drug, basically Aspergillus niger. So penicillin are classified into three main types. First is natural penicillin, second one is semisynthetic penicillin, and third one is beta-lactamase inhibitor. So natural penicillin, it includes benzyl penicillin, which is also known as penicillin G. Semisynthetic penicillin is again classified into three types, that is acid resistant, penicillinase resistant, and extended spectrum. So acid resistance, the semisynthetic penicillin, it includes phenoxymethyl penicillin, which is also known as penicillin V. Penicillinase resistance, example is penicillin and amoxicillin. Extended spectrum are again classified into three types: aminopenicillin, carboxypenicillin, and uridopenicillin, which is basically depends upon their chemical nature. In the aminopenicillin, the amino group is present. Carboxypenicillin, carboxyl group is present. And in uridopenicillin, the uracil group is present. So the aminopenicillin, this particular antibiotic, is active against Haemophilus influenza. Carboxypenicillin, this particular antibiotic, is active against Klebsiella and Pseudomonas. And last one, uridopenicillin, this particular antibiotic is active against pneumonia. And the last class, that is beta-lactamase inhibitor. The most common example is tazobactam. Tazobactam, it is the beta-lactamase inhibitor which is responsible to inhibit the cell wall of that particular bacteria. Next, there are three adverse effects of penicillin antibiotic. First, that is local irritation. The inflammation or infection occur in that particular body part due to the chemical agents such as soap, detergent, acids, alkali, some microbial agents such as insect bite. So that is the local irritation. The symptoms of local irritation, that is swelling, then itching and redness. Second one, that is a hypersensitivity reaction. The autoimmune diseases are nothing but the hypersensitivity reaction response. So hypersensitivity reaction, excessive or abnormal immune response against that particular antigen. There are four types of hypersensitivity reaction: type 1, type 2, type 3, and last one, type 4. The type 1 hypersensitivity reaction, it is the immediate response, which is also known as IgE mediated immune response. So this particular antibody, that is IgE, which is the immunoglobulin. So this IgE binds to the mast cell and there is the release of histamine. Type 2 hypersensitivity reaction, which is also known as IgG or IgM mediated immune response. There is the activation of complement system. Either it is classical, either it is lectin pathway or the alternative pathway. Type 3 hypersensitivity reaction is also known as antibody mediated response. So antibody mediated response means there is the formation of antibody and antigen complex. And the last type, that is type 4, it is known as cell mediated immune response. So T cell are responsible to release cytokine. That response is nothing but the cell mediated immune response. the most common adverse effect, that is hypersensitivity reaction. Third adverse effect, that is Jarisch-Herxheimer reaction, which is nothing but the inflammation occur in that particular body part. So these are the adverse effect. Next antibiotic, that is cephalosporin. Cephalosporin, it is also the beta-lactam antibiotic. It is isolated from the fungi, that is Cephalosporium. It is divided into 5 generations on the basis of spectrum. Either it is narrow spectrum or broad spectrum. So first generation, second generation, third generation, fourth generation and fifth generation cephalosporin antibiotics. The first generation antibiotic are active against gram-positive bacteria. Second, third and fourth generation antibiotic are active against both gram-positive and gram-negative bacteria. And the last generation, that is fifth generation antibiotic, it is active against MRSA. MRSA, stands for methicillin resistant Staphylococcus aureus. Next antibiotic, that is imipenem. Imipenem, it is an intravenous beta-lactam antibiotic discovered in 1980s. It was the first member of carbapenem class of antibiotic. So this carbapenem class of antibiotic, it is active against beta-lactamase enzyme. And this particular beta-lactamase enzyme is produced by using multiple drug-resistant gram-negative bacteria. And this carbapenem antibiotic is effective against the treatment of various types of infections. Next, it shows the broad spectrum activity, that is active against both gram-positive as well as gram-negative bacteria. But it is particularly important for its activity against Pseudomonas aeruginosa and Enterococcus species. But it is not active against MRSA. The cephalosporin, it is active against MRSA, but the imipenem, this particular antibiotic, is not active against MRSA, that is methicillin resistant Staphylococcus aureus. Imipenem antibiotic is also known as Premaxine. The chemical formula of imipenem, that is C12H17N3O4S. Next antibiotic, that is meropenem. It is also the beta-lactam antibiotic, but it is the injectable antibiotic. It belongs to the subgroup of carbapenem. It also shows the broad spectrum activity against gram-positive bacteria, gram-negative bacteria, aerobic as well as anaerobic bacteria. It was first marketed by AstraZeneca under the trade name, that is Merem. The chemical formula of meropenem, that is C17H25N3O5S. What is the mode of action? So the mode of action, that is a bactericidal, killing mode of action. But there is one exception, that is Listeria monocytogen. It shows bacteriostatic mode of action, that is inhibitory mode of action. So these are the trait names include Merocin, Merobat, Meronem, Merocil, Merofit. So Merofit, it is present in India. And the last one, that is Merosa. These are the trait names of meropenem antibiotic. Next antibiotic, that is piperacillin. It is also the broad spectrum antibiotic. It belongs to the uridopenicillin class. Then it is active against gram-positive bacteria and gram-negative bacteria. Basically, the side effects of piperacillin, that is diarrhea, constipation, nausea, headache, and insomnia. It is also known as anti-pseudomonal penicillin. The mode of action of that particular antibiotic is this particular drug inhibits the bacterial cell wall synthesis by binding on penicillin binding protein. So these are the drug receptors which are present on the surface of particular bacteria. And it also inhibits the final step, that is a transpeptidation, that is the cross-linkage reaction. The last antibiotic, that is tazobactam, which is the beta-lactamase inhibitor, which is responsible to inhibit the cell wall synthesis. It is a pharmaceutical compound that inhibits the action of bacterial beta-lactamase. It inhibits the enzyme, that is beta-lactamase, which is also known as penicillinase. Then it also shows the broad spectrum activity against gram-positive, gram-negative, aerobic as well as anaerobic bacteria. It is most commonly active against the Pseudomonas aeruginosa. Next, we see the production of penicillin. So by using aerobic batch fermentation. Here the first step, that is selection of microorganisms. So we identify the most suitable strain which is responsible to produce the most desired product. That is the selection of microorganism. We use penicillin, Penicillium chrysogenum, Penicillium notatum as well as Aspergillus species. Basically Aspergillus niger species, it is the most selective microorganism for the production of penicillin. Next, we prepare the media, that is fermentation media for the penicillin. So this particular media contains corn steep liquor, soya meal, whey, carbon source, nitrogen source, molasses, then phosphate buffer with calcium carbonate, that is CaCO3. So this phosphate buffer with calcium carbonate is responsible to adjust pH level up to 5 to 6. Also we add the precursor molecule, that is phenyl acetic acid. Then we add some nutrients such as zinc, magnesium, potassium. So these are the media preparation. Next one is strain improvement. So strain improvement basically by using the mutagenesis and by using UV radiation, we improve or develop the particular strain of that particular genus which is responsible to produce the desired product. So mutagenesis, that is changes occur in genetic material present in that bacteria. Next step, that is fermentation process. So for the fermentation process, there is the incubation period. So incubation period, that is up to 7 to 9 days. pH we maintain, that is 5 to 6, and temperature, that is 25 to 30 degrees Celsius. So the fermentation process is carried out by using surface fermentation process and submerged fermentation process. Last step, that is recovery of product. So under the recovery of product, first we separate the cell by using centrifugation and filtration technique. So there is the removal of microbial cells. Second, we add some chemical compounds, that is butyl acetate and potassium. Butyl acetate and potassium added to that particular filtrate. Then due to that chemical, there is the precipitation occur. Then we filter this particular solution by using the filtration techniques, and Then we wash the filtrate. Next step, that is purification, which is the most important step during the production process. Purification basically done by using chromatography technique, that is HPLC, high performance liquid chromatography. Then crystallization. Also we use the extraction methods. The penicillin rich aqueous broth, there is the addition of activated charcoal. So this activated charcoal is responsible to remove the pigments and impurities present in that particular solution. So this purified form of penicillin is identified by using TLC, that is thin layer chromatography, and the visualization is done by using UV illuminator. Next, the sterilization occur, both the batch sterilization methods as well as continuous sterilization methods. Quality control is done by using well diffusion method. And the last step, packaging. So this is all about the antimicrobial drugs or antibiotics. Thank you.